ആശംസകൾ നേരുന്നു. പ്രത്യേകിച്ചും ഡോക്ടർ സുനിൽ സാറിൻ്റെ ആദ്യത്തെ ഡിഫൻസാണ് ഇതിൽ വരുന്നത്, സ്റ്റുഡൻറ്റിൻ്റെയാണ്. വിവേകിൻ്റെ ഒത്തിരിയേറെ ആർട്ടിക്കിൾസ് ഇതിനകം തന്നെ പത്രങ്ങളിൽ വാർത്തകളായിട്ട് വന്നിട്ടുള്ള ഈ അവസരത്തിൽ നന്ദിയോടുകൂടി ഓർക്കുകയാണ്. സോ വിഷ് യു ഓൾ ദ ബെസ്റ്റ്. അത് നൈസ്. താങ്ക് യു.
The proceedings of today's program are controlled by the committee with the following members: Dr. G. Madhu, Professor Emeritus, Division of Chemical Engineering, CUSAT, who is the chairman; Dr. Subin K. Jose, Assistant Professor and Supervising Teacher, Department of Geology and Environmental Science, Christ College, Eringalakkada, who is the convener; and Dr. Father Jolly Andrews, Principal in charge of it and the head of the institution, Christ College, Eringalakkada. Now, I hand over the mic to the convener, Dr. Subin K. Jose, to take over this.
Good morning to one and all. Respected Principal Father Jolly Andrews, respected Manager Joy P. Today's convener Dr. G. Madhu, teachers, research coordinator, students, a warm good morning to one and all. Now we have a Ph.D. open defense of Mr. Vivek Chandran on the topic Ecology and Diversity of Ordinals of Central Kerala, India. It's under my guidance, it's a first Ph.D. Now I invite Dr. G. Madhu, the Professor, School of Chemical Engineering and former Director, School of Engineering, and my beloved research gate. Please take the next.
Good morning to all of you, respected principal, respected manager, esteemed faculty members and students. We are assembled here for the open defense and viva voce of the Ph.D. thesis submitted by Mr. Vivek Chandran A. The title of the thesis is Diversity and Ecology of Ordinates in Coal Wetlands, Central Kerala, India. Anyway, I am not going into the details of this thesis. This is a significant work. In fact, this study is the first of its kind in the state of Kerala, especially with respect to freshwater ecosystems. So, may I request Mr. Vivek Chandran to make a brief presentation of his findings. It may take about 45 minutes. About 45 minutes for the presentation, and it will be followed by discussion. So, over to Mr. Vivek Chandran. Thank you.
Good morning, everyone. Thank you for attending my Ph.D. thesis defense. So today I will be defending my Ph.D. thesis titled Diversity and Ecology of Ordinates in Coal Wetlands, Central Kerala, India. I did my research under Dr. Subin K. Jose of the Department of Geology and Environmental Science here in Christ College of Eringalakkuda. So to briefly talk about ordinates, we are mostly dragonflies and damselflies. They belong to the order insect order Odonata. So the Greek term odonto means strong mandibles. So these are the insects with strong mandibles, strong biting and chewing mouthparts. So they prey on smaller insects, and mostly they fall into two groups, that is supporters, Anisoptera and Psychoptera. Anisoptera are the dragonflies that we are all familiar with. If you look at the term Anisoptera, Aniso means unequal and Ptera means wings. So dragonflies will have unequal wings because their hind wings are broader compared to the forewings. But in the case of damselflies, that is, they fall under the supporter Psychoptera. You must be familiar with these insects. They are small and slender, and all of their wings are of equal size and shape. And there is a third group, that is Anisopsychoptera, which falls in between the Psychoptera and Anisoptera. That's a very rare group. Only 3 species are found in the world today. Only one species of this in India, that too in Darjeeling Himalayas. So we are not concerned with that in our study. So our study was focused on the diversity and ecology of anisoptera and cytoptera, that is dragonflies and damselflies, seen in the cold wetlands. So as you know, the dragonflies and damselflies are semi-aquatic insects. So they require fresh water to lay their eggs, and their larvae hatch out, and the larvae grow by molting multiple times. They shed their exoskeleton and they grow. And for the final instar larva, for molting, it comes out of water, and the adult emerges out straight from the final instar larva. So the metamorphosis here is hemi-metabolous development, which means there is no intervening pupal stage. So in the case of butterflies and other insects like beetles, you would have seen there is a pupal stage where the insect rests, but there is no pupal stage here. So it's a hemi-metabolous development because these are ancient insects. They originated on the face of Earth some 300 million years ago. So the same kind of lifestyle is maintained. And here at the top you can see a dragonfly larva that looks like a spider and is well hidden. So it is an ambush predator. It will wait for other smaller insects inside the water body. And once one unsuspecting victim comes here, it will just grab onto it and feed on it. So at the bottom you can see a damselfly larva, and from the picture you can identify it as a damselfly larva because just like the adult, even the larva is very slender. And at the tip of its abdomen you can see 3 appendages. These are caudal lamellae used for breathing. But in the case of dragonfly larvae, these cannot be seen. They have internal gills for respiration. To talk briefly about their morphology, like any insect, the automate will have a head, thorax and abdomen. And the head is almost 80% of the head is covered by 2 large compound eyes, so they are highly visual animals. So they hunt by their vision mostly. So they have very short antennae, unlike other common insects like cockroaches, because their sense of olfaction is very less. And the thorax has strong flight muscles, and they are one of the strongest flyers known in the animal kingdom. So the thorax will have 2 pairs of wings and 3 pairs of legs, and the legs are very spiny, used for grasping prey in the air. And the abdomen you can see is very small and slender. It is slender and long, and it is made up of 10 segments, which are like the compartments of a train. And at the tip of the abdomen, you can see something known as like finger-like appendages called caudal appendages or anal appendages. They are well developed only in the males because they are used for grasping the female while mating. So in the females, it is not well developed. So these are the body features depending on which their taxonomy is based. Now their role in the food web is mostly as predators. The adults mostly feed on flying insect prey, including mosquitoes and flies. These are mostly pests, and in that way they are beneficial to humankind. And the larvae mostly feed on aquatic invertebrates, sometimes even vertebrates like fishes and tadpoles. So the larvae has something known as the labial mask, which is a specialized organ seen only in the larvae. So it is, as you can see, it can be protruded out, and using which you can capture its prey and eat on it. They are also prey for a lot of animals in the ecosystem, like mostly spiders and birds. Some of the birds even plan their journey, their transcontinental migration according to the availability of dragonflies. So they have a very important role to play in the ecosystem. And they are also natural pest controllers and farmers' friends because they feed on a lot of agricultural pests. So in this picture you can see a coromandel knotfrog. It's a very common damselfly preying on a pest of rice. And they are also good bioindicators because of the following reasons. Because they are very colorful and easy to identify. Any kid, even a school-going child can actually, with the help of a field guide, he or she can identify the species. And they show only moderate species diversity. If I were to study some other group, like let's say beetles in an ecosystem, there will be thousands of species. But in the case of damselflies, if you go to a wetland ecosystem, there will be 50 to 60 species. So they are easy to monitor, and they have short life cycles. So in the tropical conditions, our ordinary species will have more than 1 life cycle in a year. So from egg to larva to adult, there will be multiple times it will keep repeating within a year itself. So if there is something happening in the environment, something drastic happening in the environment, it will be reflected in their cancer numbers very soon. And they spend a major part of their life in water, in freshwater bodies. So we are also dependent on freshwater bodies for our daily activities. So if there is something wrong with the freshwater bodies, it will easily be reflected in their diversity. And they have strong response to human disturbances, including pollution, deforestation, and climate change. So the question is what to monitor. So there are different life stages of ordinates that we can study, but eggs are too small. They are maybe tiny and they are not classified properly, so we cannot depend on that. But each of the other life stages, including adults, larva, and exuvia, they have their own advantages and disadvantages. If you are studying adults, then it's very easy. They are easy to observe and study. But in the case of larva is this life stage that is directly inside water, and it gets affected by the water quality. So, but the disadvantage with the monitoring the larva is that it is very difficult to collect and sometimes also hard to identify. Next stage is exuvia. Exuvia is the exoskeleton left behind by the final instar larva once it emerges. So using that we can identify to the species level, and the presence of exuvia in a freshwater ecosystem shows that the ordinary species has completed its life cycle, entire life cycle there. So an adult has come, laid its eggs, larvae have developed, and it has developed into an adult. So the entire life stage is completed there. So the exuvia shows that the habitat is very suitable for the species. But the disadvantage at that stage is that it is very hard to get. Some of the exuvia will be deep inside water bodies, and some will be blown away by the wind. So in this study, we actually incorporated all these life stages and took the study forward. And the study was conducted in wetlands. Wetlands, as you know, are dynamic ecosystems. There is periodic rise and fall in water, and it is it is dominated by aquatic vegetation, right, and hydric soils. The core wetlands spread over the two districts of Thrissur and Malappuram lies parallel to the west coast of India. So the paddy cultivation had started here some 300 years ago. There are presently more than 130 parcel committees. These are farmers' collectives which manage the farming activities in the wetlands. Some 40% of Kerala's rice production come from the core wetlands, because this is a very productive ecosystem. Because the two rivers Kecheri and Kerala, which flow from western Ghats, bring in a lot of alluvium. So this is a very profitable business. The paddy cultivation is still profitable in these parts of the state. And this lies below sea level, and there is seasonal inundation of water. Now we look at briefly the ecosystem services provided by this wetland. So we humans directly depend on these wetlands for our food, mostly as paddy and also as fish. A lot of fish is being caught from here, and it helps to recharge the groundwater. It absorbs a lot of water and makes water available to the nearby wells and ponds. And when in the rainy season, when there is a lot of rain, they act as a sponge and absorb the excess water and prevent flooding in the nearby areas. And it has an immense aesthetic value. If you go to these wetlands, you can see humans enjoying the nature, cycling and flying kites and all. And it also has an immense tourism potential that is largely untapped. So sites like this, beautiful sceneries like this are not uncommon in these wetlands, especially during early mornings and late evenings. It is also rich in biodiversity, especially in the case of birds. It is one of the three Ramsar sites in Kerala. The other two being Shastamkota and Ashwati Lake. So it is rich in biodiversity. It supports over 20,000 water birds in the migratory season, and there is more than 1% global population of multiple water bird species here. But in spite of providing all these benefits to human communities, wetlands are threatened because of various anthropogenic pressures, including encroachment, as you can see here. And the ordinary diversity of these wetlands was not explored before the study. Even a checklist of the ordinary trees in this wetland was not available before we conducted the study. And with this being, the ordinates can actually act as indicators of the human disturbances, despite which the studies were not happening here. So that is the background of this study. So the major objectives of the study was to assess the diversity of ordinates by monitoring their adults and exuvia. The second was to compare the diversity between these wetlands and nearby ponds. And the third was to study the ecology of ordinata larvae. And the fourth was to taxonomically describe it. Till this point of time, some of the life stages of the ordinates were not described. So we also managed to describe some of the life stages. So this was mostly an observation-based study where we collected samples only when it was absolutely necessary to describe the adults of the larvae. We had 9 long-term study sites spread across the length and breadth of the wetlands. So we used binoculars and cameras mostly, but insects were collected only when it was necessary to describe them. So we used fixed transect, visual encounter surveys, and quarter sampling. Quarter sampling was used for monitoring the larvae. So the water quality parameters were measured using a multi-parameter water quality analyzer that we, it was tested in the lab and calibrated and taken to the field. And other parameters were obtained from the nearby weather station at Kerala Agricultural University in Eringalakkada. Pesticide residue was also tested in the lab of Kerala Agricultural University. And the softwares mostly used in the study are QGIS for drawing maps and RStudio for statistical analysis. So the entire thesis is organized into 6 chapters. The first chapter, that is this chapter, is an introductory one where I introduce the audience to the dragonflies and dancerflies and the habitat here. And the next 4 chapters deal with the 4 objectives. And the 6th chapter is the recommendations for conservation of core wetlands and the future studies. So now we come to the first objective, where we study ordinary diversity of these wetlands by monitoring the adults and exuvia. The background is that there is a consensus among scientists that is a global insect decline is happening because of anthropogenic pressures like agrochemicals and habitat loss. Most of the insects in the world are threatened and the numbers are collapsing. So So what happens is that they form the base of the ecosystems. So if the insects go, then the ecosystem services that these ecosystems provide will also be lost because this ecosystem collapse is imminent. So it is very important to understand how the insect populations are faring in and around our wetlands and other habitats. That is the background of the study. And here you can see the wetland. There are, the entire wetland is sandwiched between two rivers, the Kecheri, not the Kecheri. At the top you can see the Bhagapura, and at the bottom you can see the Chalapuri river. So the entire stretch of wetlands is sandwiched between these two rivers, and it is drained by Kecharipura and Karavenu river. So we had actually three divisions within the wetlands. Between the Bhagapura and Kecharipura, we had the Ponnani Code, and between Kecharipura and Karavenu river, we had the Thrissur North Code, and between Karavenu river and Chalapuri river, we had the Thrissur South Code, mostly in Eringalakkada and nearby regions. So we had three permanent study plots in all these three locations, which were supplemented with randomly visited sites also. So we went to the field between 9 AM and 11 AM when the ordinates are mostly active because they are poikilotherms, they are cold-blooded animals, so they need the warmth of the sun to warm themselves up, and they are mostly active during the 9 AM to 11 AM. That's when the field visits were done. Full bit well transects were done in these places, and the study was carried out for 2 years, and in each year there were 4 seasons. Monsoon 1, Monsoon 2, winter and summer. So in each year I visited the same field 4 times, and I collected the exudate while walking them. So the result is that we were able to record a total of 63 species of ordinates within the 2 years. Out of which 3 species are endemic to the Western Ghats region and 2 species are endemic to India. These are the ordinate species that we were able to record. I'm just quickly running through them. All the species were photographed from the cold wetlands. And this is a list of the species that we were able to record. So this lists all the scientific names of the 63 species that we were able to record from the wetlands. And regarding exuviae, we were able to collect 58 exuviae belonging to 19 species. So if you compare with the 63 species, it is very less. So either of the two is true. Either not all species are able to complete their life cycle there. That is why we were able to get only the exuviae of 19 species. Or maybe the sampling exuviae is not the right way to monitor ordinates, because some of them will anyway be lost because of the winds and other parameters. So that is what the collection of the exuviae and the study shows. But if you look at the graph, we use Shannon diversity index to monitor the diversity of ordinates here. And you can see that the Shannon diversity index is, in both the years, it reaches the maximum during the early monsoon months. There is a slight dip during the late monsoon months, and there is a immense dip. There is a dip during the real dip during winter months, and it slowly picks up during the summer months. So the same is true across the 2 years, and you can see by the size of the dots, you can see that the most number of exuviae were collected during early monsoon period. So that is the period when the adults are emerging from the larval stage. That is what the graph shows. And regarding the parameters of weather, total rainfall, relative humidity and wind speed were positively correlated with the ordinate diversity, but the effect of temperature was not clear from the study. Now this is the most abundant species that we were able to record from the wetlands. This is the Pandala flavescens. It is known as the globe skimmer because it is seen throughout the globe, and it is a migratory species. It is seen almost in all parts of the country and of the world, and it travels according to the monsoon patterns. So counting them was, we had to modify our methodology a bit because we were using full well transit. Using it was very difficult to study them. So we did this. After doing the well transit, we went to a spot and we went to a vantage point and did photographs and did a block count after putting the grids. So you can see that their numbers were reached the peak in both the years. Their numbers reached the peak in the late monsoon months. And there are different kinds of aquatic vegetation seen here, mostly floating, free-floating ones and floating rooted and emergent types and submerged types. So each transit had its own major aquatic vegetation type, and the species accumulation curve, we can judge that the free-floating aquatic vegetation type had the highest species incidence of ordinates. And also we can see that the graphs are flattening out, which shows that our sampling is almost complete. We also did a statistical analysis to check if the ordinate diversity differs significantly based on the aquatic vegetation. So the result of the Permanova, this permutational multivariate analysis, is significant. You can see that the p-value, in the second table you can see that the p-value in the case of vegetation type is 0.003 less than, which is less than, p is like 0.05. So it is significant, and the aquatic vegetation type is having a significant impact on the ordinate diversity, and this then free floating vegetation is able to support maximum number of species, and submerged vegetation is only able to support only the minimum number of species. So the takeaway from this study is that cold wetlands have immense conservation value because it supports over 30% of the species seen in Kerala are seen here, and many of them are endemics, including resident endemic species. And there are clear seasonal patterns where the adults are emerging during the early monsoon months. And the exhibit sampling also revealed that 19 species, at least 19 species, are completing their entire life cycle in this wetlands. So the weather parameters, while monitoring them, we were able to see that rainfall, humidity, and wind speed are having a direct correlation, the positive correlation with the ordinary diversity. And aquatic vegetation that is also having an impact on the type of ordinary communities in the location. For the next study, we compared the ordinary diversity of wetlands and ponds. This was also to see if the ponds are able to function as surrogate wetlands. It is able to support as many species as the wetlands can. So for this study, we used a rapid method. From the previous study, we had concluded that the adult ordinary diversity was highest during the early monsoon months, that is May to August. So that is when this study was conducted across the year of 2021. We selected 20 wetland sites and 20 pond sites. So all these 20 sites were located in the Thrissur cold wetlands, that is South Cold wetlands, below the Kairenu river, and all the ponds were located near Eringalakkada, almost of the same landscape. We also measured the water quality parameters and tried to correlate how the Kodakata community differs according to the water quality. So the results show that wetlands are able to support more number of Kodakata species, definitely. Because we were able to record a total of 44 species of organisms from the wetlands, and 14 of them were exclusive only in the wetlands. They were not seen in the ponds. But in the case of ponds, we were only able to record 30 species, and it had no exclusive species, which means all the species seen in the ponds were also seen in the wetlands. And this is a correlation matrix which shows how the water quality parameters are related to each other and also to the species richness. From this, you can conclude that the conductivity, turbidity, and TDS, you see, it is having a negative impact on the adult ordinary diversity. Next, we proceeded with the multiple linear regression to see which of the water quality parameters are driving, having the maximum effect on ordinary diversity. And we found that dissolved oxygen is having the most positive effect on the ordinary diversity, and salinity is having a negative effect, which was expected because they are freshwater organisms and they are not able to tolerate the salinity. This was a validation test done to check if the multiple linear regression is valid. So since it is the histogram of residence is showing a bell curve, that is a normal curve, we can conclude that our model is valid, and that these two are the dissolved oxygen and salinity were the most important factors driving adult ordinary diversity in these ponds and wetlands. So we also did a Karanova for this vegan package was used in art using Euclidean distance, and we also found that the habitat types, they had the ponds and the wetlands had entirely different kind of water quality parameters. So if you look at the graph below using NMDS, that is non-multidimensional scaling, the same result of the table is being shown in a graphical manner. You can see that all the wetland sites which are shown in blue, they are clustering together, and all the red dots are the pond sites. They are clustering differently from the wetlands. So this is because of the different water quality profiles in the two habitat types. So if you look at the same graph here, we have actually superimposed vectors here. So you can see the blue dots these are wetlands. They are mostly regulated with they have high POD content, high nitrate content, and temperature and salinity. Whereas in the ponds, it is driven by high TDS, conductivity, turbidity, and phosphate. This is because of the different land use. Because in the ponds, mostly people use the ponds for washing clothes, and they use a lot of detergents. They may also take baths there. They may wash their clothes there. So a lot of phosphate input is there, which is also causing turbidity to go high. But in the case of wetlands, there is a lot of nutrient input, mostly in the agricultural area. So that is why the BOD and nitrate levels are high here. So the same is being the NMDS plot that is being shown in the tabular form here. And we also did another Karanova to test if the ordinary community differed significantly between the habitat types. If the ponds and wetlands had different ordinary community. So you can see that the p-value is less than, again, p is less than 0.05, this p is equal to 0.001, which is highly significant. So the ponds and wetlands are definitely having different ordinary composition. And we did a simple analysis, the similarity percentage analysis, and according to it, the Pentala flavescens and Trichodermis contaminata, these are cumulative contributions of the species. These two species are driving the difference between the ordinary communities in the two habitat types. Next, we ran an indicator species analysis using the water quality parameters. So here you can see, the statistically significant results are displayed here. Each species has a different, response to the water quality parameters. So different species will have different responses to the same parameter. If you look at the table, the Eringalakkada Christ, which is a small brood dragonfly, a very common species, it is having a positive relation with nitrate. So it is able to tolerate nitrate and thrive in water, waters where nitrate is abundant. But in the case of Eringalakkada, which is a small species, small damselfly species, it is not able to tolerate nitrate because it is a sensitive species. So we have identified 8 species which are potential indicators of different water quality parameters. So we ran the same indicator species analysis according to habitat type also, and we found that 3 species are, they are having strong preference for wetland habitat type, but none of the species were associated with pond. So none of the species showed any preference for ponds because all the species seen in ponds were also seen in wetlands. But these 3 species definitely require wetlands. They are not able to survive in the ponds. So to conclude, we have compared the wetlands and ponds and found that the water quality profiles are significantly different between wetlands and ponds, and the Odonata community are also different. So ponds are not able to act as surrogate habitats for odonates. So wetlands have to be controlled. There is no other law because ponds are relatively undisturbed because we all of the the wetlands are mostly used by farmers and the limited community. So they may get filled up, but we think since the ponds are around, we are able to conserve most of the biodiversity. But no, ponds are not able to conserve the biodiversity because many of the species require wetlands. That's the takeaway from this chapter. And next we come to one of the most important chapters of this thesis, that is the ecology of ornamental larvae. I say so because in India there are not many studies focusing on the ecology of ornamental larvae. Because most of the studies are focused on adults. We do not know how the larvae survive underwater and what are the parameters that control the distribution. So for this we use the quadrant method. The standard method was to use a 1 meter into 1 meter quadrant, which was put and the water quality, the vegetation parameters and soil parameters were monitored along with the larvae. The larvae were collected and identified in the field itself, but if they could not be identified in the field, we should take it to the lab and rear them here. And once they are emerged, we base the identification on the adults, and then we discard the larvae. So from this study was carried out across again the 4 seasons in a year, but we were able to carry out the study for only 1 year. For the adults and exhibit, we did the study for 2 years, but because of logistical reasons, we were able to do the study for just 1 year because there were a lot of parameters to be monitored and the larvae had to be reared indoors, so that is a lot of task. So if the species accumulation curve shows that it is flattening out, the species accumulation curve is flattening out, but if we continue the study for maybe a few more years, we will be able to collect more larvae, more species. So the species that we were able to collect belong to 33 species. So out of the 63 species seen as adults, we were able to collect the larvae of only 33 species. And out of which the Agriopinus pygmea, you can see that is a small damselfly, that is a larva that dominated this ecosystem. And if you look at the seasonal data, we can say that this is exact ulta or the opposite of the adult diversity. Adults were more, the species richness and their diversity index was more during the early monsoon months. But in the case of larvae, the early monsoon months had their minimal numbers, and the number of larvae and their diversity was highest during the late monsoon and winter period. So now a gradual picture is emerging. So we have monitored the exuvia, and by monitoring exuvia, we know that most of the exuvia are found during the early monsoon months when they emerge, and the adult diversity is also highest during the early monsoon period. And during the late monsoon and winter months, the larval abundance is more, but the adult diversity slowly crashes, and the number of larvae gradually picks up during summer, and the adults emerge during the next monsoon. So we, by observing the exuvia, larvae and adults, we are able to piece together the entire life cycle of odonates in the wetlands. This is a correlation heat map of all the parameters monitored in the study, and it shows that you can see that the nitrate and phosphate levels are actually having a positive impact on odonata diversity. So that is mostly because it is not the enrichment of the nutrients here is not leading to hypoxic conditions. That is because of mixing of water. So generally, if there is a lot of nutrient input, there will be eutrophication happening. A lot of weeds will grow, but that is not mostly happening in wetlands as the study shows, because there is a lot of mixing of water mostly because of the agricultural activities. So the farmers pump out the water from the wetlands into the canals for the cultivation, and they also pump the water back to the fields when the water is needed. So there is a lot of mixing of water, and that's the reason why the water is not turning eutrophic here. And we tested for normality using the quantile test. And you can see that most of the parameters are aligning with the diagram. This shows that all the parameters are mostly normally distributed, and we proceeded with principal component analysis, again with the vegan package of RStudio. So this was run to test, which of the 20 parameters monitored are having the strongest influence on ordinata diversity, especially larvae. So we can see that here the PC1, PC2 and PC3, these are the principal components. Principal components 1, 2 and 3 together contribute to over 49% of the variance. See, the growth of ordinata larvae there. And the summer is, that summer is not a very good time for ordinata larvae because the water levels are less. And also there is a mixture of the conditions of all the other seasons. So these are in this study we were able to conclude which are the water quality and soil and vegetation parameters which are driving the ordinata larval diversity in the cold wetlands. And we end this chapter by saying that conservation strategies should prioritize a heterogeneity in habitats because different species require different habitat features for their survival and conservation. Now we reach the next chapter. This is the final objective, to taxonomically describe the life stages of ordinates that were unknown to humankind till this point of time. Because any ecological study is based on taxonomy. If your taxonomy is wrong, then you cannot proceed with your ecological proceedings. So for collecting adults, we used a butterfly net or an aerial net, using which adults were collected if we were not able to identify them in the field. And we brought them to the lab and studied them under the microscope, and descriptions were given. And for larvae, some of the larvae, especially the smaller larvae, we were not able to identify in the field. So such larvae we brought to the lab and again read them in the laboratory and based the identification on the emergence of adults. And once the adults emerge, all the larval features are visible in the exudate itself. So using the exudate, we describe the larvae. So the first description was of the Macrorhopus minardicus. This is an endemic species seen in the Western Ghats. It was described using only female specimen by the British scientist Fraser in 1924. So after almost 100 years, we were able to describe the males from the cold wetlands. We collaborated with the scientists of Kanyakumari and Goa for this. They had also collected specimens from there, and we made the comparisons and studied and published this in the journal Ornithologica. So it is a very large species, it is a very seasonal species, and according to the IUCN release, this data deficient, not much is known about the species. So they were to give the first description of the species. Next we proceeded with the description of the larva of the Platyhelsis platycellus. This is again a very rare species. Out of the 163 larvae that we collected in the study, we were only one belonging to the species. So the Platyhelsis genus has only 4 species in the world, and none of them had the larva described. So we read the larva inside and read it to the adulthood, and we were able to describe the larva. So you can see in the sea, you can see that it is the labial, that is the labial mask described. It is a very peculiar for this species. It is very spiny. It is used for catching its prey. And below you can see the pictures that is high level in the cold wetlands, that is Ponani Cold, from where the species was collected. Next, this is a very common species actually, but the larva was not known. It is very common in the Indian subcontinent, in all the wetlands and ponds, but the larva was remained unknown till our study. So we described based on the larva that we read in the laboratory. So this species had a short labium, as you can see in the B figure, and it had abdominal spines which protects it from the its predators, and its body was covered with minute setae which trap sediments and help it camouflage itself. It remains hidden from its enemies. So in this chapter we were able to describe the species, some of the descriptions that were not available scientifically, and we hope that this will take the ornithology of in India forward, because some of these are the very common species, but some of the life stages were undocumented. So if we have to use them as indicator species, we need to first be able to identify them as a species. So for that, we hope that this chapter will help. And now we come to the recommendations. Throughout the study, we were able to see a lot of invasive plants here. So that is especially the invasive, that is submerged plants called Kabamba is happening. In our study, we were able to conclude, if you remember the species accumulation curve, this aquatic vegetation type, that is submerged type of vegetation, was having an adverse effect on the ornamentum. So this is definitely having an impact on the native biodiversity, and they must be managed properly. They should be collected and removed, and native vegetation should be let to grow. And there is also effect of invasive fishes. We were able to come across a lot of invasive fish species during the study, but that is not part of the study. That is a limitation. That has to be, such studies should be carried forward. And we were also able to get some pesticide residues. So So if the pesticides were being used in the agriculture according to the law, then the residues should not be available in the environment, but we were able to come across the residues of the pesticides, fenvalerate and bifenthrin. So these should be managed properly. And future research directions, we have identified some species as indicator species in the study. These are potential indicator species. They are studied in the wild. Now they have to be brought to the laboratory, and the study should be experimental. And it should be concluded which species can be used as indicator species. And in the cold wetlands, we get a great abundance of organisms. A lot of dragonflies and amazonflies can be seen, and we can study their behavior and population dynamics here. And there are other lot of organizations studying ordinary diversity and other diversity of other organisms of these wetlands, but they mostly work in silos. Even here we have zoology department, we have environmental science department, but we were able to collaborate and come up with a paper. So such like that, we should have collaborations with sister institutions and take the studies forward, because the resources are limited. So if we are able to share our resources and knowledge, we will be able to take the studies on these wetlands even further. So these are some of the papers that we were able to publish. Most of the papers were published in Ornithologica and Syntaxa and Journal of Certain Taxa. These are some of the presentations we gave in the national and international seminars. These are some of the important references, and I have also kept the raw data here. Here you can see the weather parameters that were obtained from the agricultural university, and also the water quality parameters and environmental parameters measured in the study. I would like to take a moment to thank all the people who supported me and all the institutions, because I feel that I am presenting a collective study here. Even though all these people are not present here, I am representing all these people and institutions. I thank all of you, and now I am open for questions. Thank you.
Thank you, Vivek. You have made a very crisp presentation. Now the presentation is open for discussion. The audience. First one is regarding the Shannon-Wiener index, diversity index. So you have calculated this index for each transect sampling as a measure of adult ordinate diversity. So why do you set the Shannon index specifically for the purpose instead of other diversity index?
There are a lot of diversity indices available, like Simpson index, Margalus index, but we stuck to Shannon-Wiener index, also known as the Shannon index of diversity, because it is widely used in freshwater ecology studies. So for ease of comparison with other studies, so if you are doing a study in these wetlands, you should be able to compare with anywhere in the world. So for that ease of comparison, that is one of the reasons. And also that we did a lot of statistical analysis in this study. So this Shannon-Wiener index is compatible with all these studies, all the statistical analysis. So that is why, because of these two reasons. One, we should be able to compare our results with the studies happening across the world, and also with the compatibility, it had to have the compatibility with the statistical analysis. This is why we have to choose Shannon-Wiener.
Oh. As part of the study, you have carried out extensive sampling of exuvian. So, in what way did the exuvian sampling offer insights into the breeding patterns as well as the habitat utilization of ordinary species?
So, exuviae, as we know, the presence of exuviae, as we discussed earlier, it shows that the species has completed its life cycle there. So, by sampling exuviae, we were able to conclude that at least 19 species of ordinates are able to complete their entire life cycle in these wetlands. So, another has come, it has made its egg, larva has developed, and the larva has emerged into an adult. That's what the exuvia shows. So, we were able to document the life cycle of 19 species. At least 19 species were able to complete the life cycle there. And also, for piecing together the life cycle, the entire life cycle of Ordinata, this was very helpful, because we were able to conclude that most of the emergencies happened during the early monsoon period, because we were able to collect maximum number of ordinates in the early monsoon period. So that way the quantity of the exuvia were very low.
You have carried out extensive statistical analysis. For example, this permutational multivariate analysis of various Permanova. The Permanova model shows that approximately 75.62% of the overall variation in community composition. Can you explain it further?
Okay. Any statistical study, if it shows that the model is able to explain, let's say 75% of the data, of variation data, it means that the remaining 25% can be explained with other parameters that were not part of the study. So, for example, in this case, we were not studying the exotic fish. So, in the case of the adult Ordinata study that Sir mentioned here, so the 75% of the difference in the diversity across sites could be explained by the parameters studied, which included aquatic vegetation type and seasons. But the 25% could not be explained because it could be influenced by other parameters that were not part of the study, which may include the fishes and other things that were not part of the study.
So then again, everyone talks about the impact of climate change and pesticide on different types of ecosystems, especially freshwater ecosystems. Also, could you encounter any evidence of the impact of pesticide contamination and climate change affecting the habitats of ordinates?
So we were able to detect two pesticides. One was phenylate and bifenthrin. Both these are pyrethroid chemicals, are insecticides, and as I said earlier, if they were used in the quantities as prescribed by the agriculture department, none of the pesticide residues should be available in the environment. But unfortunately, in 4 samples we were able to get this, and in the sample where the concentration was maximum, we were not able to get any larvae. Even a single larva, no life form was seen there. So pesticide contamination unfortunately continues to happen in our wetlands. So that is regarding the pesticide, but their exact chronic and acute effect on the larvae can be studied in the laboratory, maybe by collecting larvae and maybe exposing them to the chemicals, how they are responding. But in the environment, definitely they are having an effect. And regarding climate change, to say about climate change, we may have to study, climate for
a long-term
study may be needed. But definitely one thing we can say that salinity was one of the negative factors influencing Ordinata diversity. So we know that with rising temperatures, there will be a rise of sea levels, and there is a threat that there will be saltwater intrusion in our wetlands. So with that, definitely the Ordinata diversity will be drastically affected.
So, what type of methodology did you adopt to assign indicator value to each species in the separate indicator species analysis?
So, there were 2 analyses run. One was for water quality and one was for habitat type. So, for water quality, we actually used the package called Indic Species. It is available for free for our studio. So, in this, what the software does is, regarding water quality, it will classify samples into high value and low value based on the median values of water quality parameter. For example, if there is pH, we are regarding pH, if the median value is 6, so whatever comes below 6 will be classified as low value, and whatever goes above 6 will be high value. And for each species documented in the study, an indicator value is assigned based on the fidelity or preference for this pH or whatever water quality parameters are studied, and a permutation test is run, and only the species that are statistically significant. If the p-value is less than, a permutation test is run, and only if the p-value is less than 0.05, the table shows the species, and we were able to arrive at 8 species which can indicate the water quality parameters.
Then you have already mentioned that salinity. Salinity has a negative effect on the growth of both the larvae as well as adult members. So how exactly salinity affects the life cycle of larvae and adults?
So it's not just about ordinates. For any species, there will be a range of tolerance for any habitat parameters. So if you take, let's say, temperature, so the minimum species may be able to tolerate is 22 and the maximum could be 30. So only if the temperature remains between 22 and 30 degrees Celsius will the species be able to survive in such habitats. So in the case of salinity, this range of tolerance is very less for ordinates because they have adapted to freshwater environments, and the range of tolerance for salinity is very less. So that is why they are not able to survive in saline conditions. They prefer only freshwater waters.
Another observation that you made was the most productive period for most of the species that you studied was this monsoon and monsoon and winter. And also you mentioned that in the early period of monsoon, the pH of the soil was very components may get leached away. That may lead to the drop in pH. It may become acidic. So in that case, anyway, that is a negative factor. If the pH drops, it may affect the growth of larvae. Can you explain this?
This happened during the early monsoon months, not early months, the late monsoon period. When the water, that is mostly in August, September, October, when the rains are more, so the rainwater is mostly acidic and the water in the wetlands mostly turn acidic. So we may think that it will have, that is the time when I collected larvae, they are small larvae. So they hide among the vegetation and sometimes they bury inside the soil also. So maybe that way they are able to shield themselves from the negative impact of pH. But in the winter months when they are bigger, the pH goes up. So when they are bigger and they can be seen easily, and when they are ready to emerge, the pH is kind of near the normal of the 7, that is near neutral levels. So maybe because they are small and they are able to hide among the vegetation and the soil, they are able to overcome this negative effect of pH.
Escape the effects of acidity. And then another observation was regarding the nutrients, nitrates and phosphates. We found that they have a positive impact. That way you have to say positive impact is the growth of larvae and the animals. But anyway, now because the nutrient content in our freshwater ecosystems is on the rise due to our agricultural practices and other human activities. It is on the rise. So naturally there is a chance of eutrophication. And the dissolved oxygen levels, there is every chance of the dissolved oxygen levels going down. So will it have some impact on this species?
Yes, definitely. In the multiple linear regression that we did, we saw that the salinity is the most negative factor, the factor that influences organic diversity negatively. But the factor that impacts it positively, mostly it is the dissolved oxygen. Dissolved oxygen is very important for them. But in these wetlands, what we have observed is that there is a high concentration of nutrients. So this might be like against our logic, but there is no drop in dissolved oxygen levels. So what I have observed is this is probably because there is good mixing of water here because of the pumping activities done by the agricultural community. Maybe this is actually beneficial for the ornaments also because there is good mixing of water and the nutrients are mixed and there is no hypoxic condition, meaning there is no, the dissolved oxygen level does not fall below a certain level. So they are able to survive. That's how maybe not just the ornaments, maybe the other aquatic life forms are also able to survive because of the agricultural activities.
So the proper mixing that takes place in the cold wetlands may
compensate for that.
May increase the biodiversity. The second adjudicator has only 2 queries from the second adjudicator. It's a general question, how does the water quality parameters influence the richness of ordinate species? The query made by the first exam.
So that is mostly a general question, but I think they have addressed it.
Already have explained.
Already mentioned. So like all species ordinance will also have a range of tolerance and water quality parameters of course affect them because that is where the larvae grow. So if there is something wrong in the water quality, there are a lot of parameters and each species will have its own response to the changes in water quality. Some may be able to tolerate low pH, some may be able to tolerate high acidity. So this differs from species to species, but since the larvae grow in water and they have a range of tolerance like any other organism, water quality affects the diversity and ecology of organisms in agriculture.
Now, in a way that we find many exotic, invasive species of plants in all these freshwater ecosystems. So how do they impact the species diversity of ornaments?
So like we discussed, most of the species seen in these wetlands are generalist species. So they are able to overcome most of the adversities that we humans, they put on the wetlands and other habitats. I've seen that the number of larvae did not drop significantly because of the exotic plants, except in the case of submerged vegetation. There is one species called Kabamba frucata that is abundant now. This is not noticed usually because when we think of exotic species in wetlands, what we think is the Kawada, that is the water hyacinth. That's also very common here, but it does not seem to have. Even the African pile that we see, Salvinia monster, it is not having an effect on the ornamental, not adverse effect anyway. Because some of the species actually prefer, because they have hanging roots, places to hide. It offers microhabitats for these species. But in the case of this species, that is the submerged species, Kabamba, it has very beautiful colors. Only the flowers will come out of the water. You might have seen this in some of the photographs. Only small red flowers will be seen from the water surface. But it is having a very negative impact on organisms because number of larvae collected and others also were not there because I think it is crowding out the native vegetation and it is not, it's like a thick mass. So maybe the larvae are not able to move around and it is crowding out native vegetation and also probably its prey. So this submerged vegetation is definitely exotic species is having a negative impact on ornaments. Likewise, the other vegetation types, exotic vegetation like Icoria may be having a negative impact on other species, not ornaments.
One final question. You have made several recommendations. Okay. So you have suggested adoption of agricultural practices that will minimize the impacts on water quality in these ecosystems. So what type of sustainable agricultural practice will you adopt?
Okay, one would be, we cannot, this is a very productive ecosystem and farming has been happening here for over 300 years. And even some of us in this room may be having land in the wetlands. So if we ask people to stop using all chemicals, that may not be feasible. But definitely we are getting pesticide residues from the soil samples. So definitely there is excessive use of pesticides. So it will actually harm your agricultural activities if you are using excessive pesticides. So what I would suggest is a integrative method to manage the pests here. Even the ordinates are beneficial to agriculture because they feed on many of the insect pests. So if you are using chemicals, use chemicals according to the prescriptions given by the agriculture department. That is one of the fine points that I would make.
Okay. Thank you, Vivek.
Thank you.
The way you responded to the queries. Okay, thank you. Now it is time to conclude this open defense session. Mr. Vivek Chandran has satisfactorily defended his thesis titled Diversity and Ecology of Odonates in Cold Wetlands, Central Kerala, India. He has also responded satisfactorily to the queries made by the audience as well as the thesis adjudicators. And as you have seen, he has a long list of publications. All these research findings have been published in peer-reviewed journals. I understand that there are 14 publications, isn't it? 14 publications. It's an excellent So, based on the satisfactory performance in the open defense and also satisfactory response to the queries and also based on the recommendations made by the adjudicators, so the board, the Viva board recommends the award of the degree of Doctor of Philosophy in Environmental Science to Vivek Chandran A, known as Dr. Vivek Chandran A. And also one more observation I would like to make. In a way, I think he has carried out extensive studies on these Odonata species, especially the dragonflies. So I think he is going to continue his studies. Maybe at one point of time, he may be known as the dragonfly man of Kerala. Great man of Kerala, and recently we have one Malayali, Professor S.T. Biju of Delhi University, is known as the frog man of India. Like that, one point of time, I wish he'll become the Odonata man or dragonfly man of Kerala. Okay, thank you very much. So we will wind up this session here. Thank you very much for your august presence in this open defense and Viva examination. Thank you.